All right, today we're going to dive deep into the very architecture of our oral health. We're talking about collagen, the single most important protein that literally holds our teeth in place. In this explainer, we're going to connect the dots between some heavy-duty molecular science and what dentists actually see and deal with in the clinic every day. So let's get into it.
We're going to start with this pretty striking image. Now, this isn't just some old photo from a history book. It's a dramatic, real-world example of what catastrophic collagen failure looks like. This is scurvy, and it shows us exactly what happens when the body completely loses its ability to produce stable collagen. The stakes, as you can see, couldn't be higher. And that really sets up the big question for us today. What's the common thread? What connects the disease that plagued sailors for centuries to the absolute cutting edge of rebuilding oral tissue? Well, the answer, as we're about to see, is hidden in the magnificent and surprisingly fragile process of how collagen is made.
So to really get why this system can fail so spectacularly, we first have to appreciate how it's built. We're going to take a look inside the fibroblast cell, which you can think of as the molecular factory that's responsible for churning out these incredible high-tensile cables that make up our periodontium.
Okay, so what you're looking at here is basically the molecular assembly line. It all starts with a genetic blueprint in the nucleus. That blueprint is then used to build these long polypeptide chains. Then, 3 of those chains twist together into that famous triple helix. But, and this is a really key detail, it's not actually finished yet. The cell packages it up and ships it outside before enzymes snip off the ends. Only then, once it's outside the cell, can these molecules stack together to form those super strong collagen fibrils. It's a brilliant safety feature, really, to make sure the whole factory doesn't just gum itself up.
So the secret sauce for that powerful triple helix? It's all in its unique amino acid recipe. The structure is this constant repeating pattern, glycine XY. Now, glycine makes up a full third of the entire molecule. And because it's the tiniest amino acid there is, it's the only one that can fit down the crowded center of that helix. Then you have proline and hydroxyproline, which create the kinks and the bonds that lock it all together. And here's the bottom line. Without glycine in every third spot, the helix just can't form, period. And that brings us to the absolute linchpin of this whole process, an enzyme called prolyl hydroxylase. Its one critical job is to turn proline into hydroxyproline. This isn't just a small change. This step creates the crucial hydrogen bonds that give the triple helix its incredible stability. It's like tightening the bolts on a suspension bridge. But here's the catch, the big dependency. This enzyme is 100% reliant on vitamin C to do its job.
And boom, there it is. That's our connection back to that sailor with scurvy. Without vitamin C, polyhydroxylase just shuts down. No vitamin C means no hydroxyproline. No hydroxyproline means no stable bonds in the triple helix. And what you're left with is weak, flimsy collagen that basically falls apart as soon as it's made. The entire structural fabric of the gums and ligaments just disintegrates. One tiny molecule holds the key to the whole system.
Okay, so we've seen the molecular blueprint. Now let's zoom out and see the masterpiece it builds. The incredible piece of biological engineering that is the periodontum. It's basically a perfectly designed shock absorber. So, take a look here. On the left, you've got the big picture of the entire tooth support system. Then on the right, we've zoomed way in to the microscopic level. See all that dense pink stuff? That is a massive, tightly woven network of collagen bundles. This is what gives your gums their firmness and their strength. It's a fabric made almost entirely of type 1 collagen.
But the real stroke of genius, the biomechanical wizardry, is right here with what we call Sharpie's fibers. These are the ends of the collagen bundles that literally embed themselves right into the cementum of the tooth on one side and into the jawbone on the other. They act like the suspension cables on a bridge, holding the tooth perfectly in its socket. And what's so incredible is that this system takes all that destructive downward crushing force from chewing and cleverly converts it into tension, a pulling force that actually stimulates the bone and keeps it healthy.
Now, the periodontal ligament isn't just made of one kind of cable. No, it's this really precise and clever blend. You've got type 1 collagen, which is like the thick, heavy-duty steel support cables that provide raw strength. But then, woven in between all of that is type 3 collagen. It's a finer, more delicate network. Think of it like an elastic safety net. This combination is what gives the PDL its unique magic. It's both incredibly strong and just flexible enough to absorb shock.
And for anyone studying for exams, here is a huge takeaway point. The PDL is not a static, unchanging structure. In fact, it has one of the highest rates of collagen remodeling and turnover in the entire body. This constant rebuilding is what allows it to adapt to everyday chewing forces, and more dramatically, it's what allows orthodontists to move teeth through bone. It's a living, constantly responsive system. So we have this perfectly balanced, beautifully engineered system. But what happens when it comes under attack? We're now entering the battlefield, where inflammation and systemic diseases can launch a full-scale assault on this collagen architecture. This table right here, it perfectly shows the shift from a state of peace to all-out war in the tissue. In health, everything's in balance, homeostasis. Synthesis equals degradation. But in periodontitis, the whole system just flips on its head. Collagen production gets shut down, destructive enzymes start running wild, and that beautiful, organized fiber network turns into a fragmented, chaotic mess. The equilibrium is completely shattered.
So who are the main villains in the story? They're a family of enzymes called matrix metalloproteinases, or MMPs. The easiest way to think of them is as a molecular demolition crew. And in the inflammatory chaos of periodontitis, one of these, in particular, MMP8, which actually comes from our own immune cells, becomes the primary weapon of mass destruction, just systematically slicing through the collagen fibers and dismantling the whole structure.
And this simple chart just lays it all out. This is the core problem. See, our bodies naturally produce inhibitors for these MMPs. They're called TIMPs. In a healthy state, they keep that demolition crew in check. But in periodontitis, the production of MMPs just skyrockets, and the TIMP inhibitors are completely overwhelmed. And it's that massive imbalance, way too much demolition, not nearly enough protection, that leads to the tissue literally being destroyed.
And it's not just about bacteria. Your whole body's health plays a massive role here. In conditions like uncontrolled diabetes, or even just as we age, these molecules called advanced glycation end products, or AGEs, start to form. And what they do is stick to the collagen fibers, cross-linking them in abnormal ways. The best analogy is that it's like putting a layer of brittle caramel or sugar coating all over our strong steel cables. It makes them rigid, weak, and much, much harder for the body to repair and replace.
Okay, but it is not all doom and gloom. Because all of this deep biochemical knowledge, we're now starting to use it to fight back, to diagnose disease much earlier, and even, excitingly, to begin rebuilding what has been lost.
So you see this number on the screen, 30.3 picograms. What on earth does that mean? Well, that's the average level of something called ICTP, a specific little fragment that breaks off of type 1 collagen found in the fluid of a diseased periodontal pocket. Now get this, in a healthy site, that number is just 1.1. So you're going from 1.1 to over 30. That's a huge red flag, and it makes ICTP an amazing biomarker that lets us see active destruction happening in real time.
And this is where the future gets really, really cool. We are now using 3D bioprinting to create custom-built collagen scaffolds. You can take purified collagen, process it into what's called a bio-ink, and then literally print a three-dimensional scaffold that is perfectly shaped to fit a patient's individual bone defect. I know, it sounds like something out of a sci-fi movie, but this is happening right now.
And here is the proof in the pudding. This is a microscopic view of cells growing on one of those 3D printed scaffolds. See all that bright red staining? That's a special dye that lights up wherever there's new mineral, new bone being formed. So the takeaway here is huge. This scaffold isn't just passively plugging a hole. It's an active structure that is successfully telling the body's own cells, hey, build new bone and ligament right here.
So let's wrap up the clinical side of things with a really simple takeaway. Just remember the 3 R's. First is recognition, using those biomarkers to spot the disease early. Second is repair, using things like collagen membranes and a process called guided tissue regeneration to basically create a space for the body to heal itself. And third, the future is regeneration, using these advanced technologies like 3D printing to actively rebuild the complex architecture that was lost.
So think about the journey we just took. We went from the level of a single repeating amino acid chain all the way to the complex battlefield inside a periodontal pocket, and now to the dawn of regenerative medicine. We've decoded the blueprint, we've mapped the battlefield, and we're building the tools to fight back. Which really leaves us with this final thought. Are we on the verge of moving beyond simple repair and becoming true architects of living oral tissue? Well, the journey is just beginning.